To know a bit about you, our audience, we have a poll and a question for you. What organization do you currently work for? And we have these options. So I'm going to launch the poll and you can answer at your ease. And hopefully we can see the audience that we have here and we can interact with you. So I'm going to launch the poll now. Let me know in the chat if you can see it or not, and if you can submit your answer. So I see there's still many people joining. We just launched a poll to get to know the audience. The session is being recorded for internal purposes. As I can see, some people already answered. We have around 44% in vaccines companies, 22% as CRO as a consultant, 22% in investigational sites, and then 11% for others. I'll now pass the word to Benedict to introduce the discovery session and to start the discovery session. And I wish you all a great learning journey. Thank you.
Well, thank you very much, Andre. And if you can just move to the next slide. Indeed, so we're very excited about the topic for today's session. We're going to talk about vaccines, and this is a hot topic given the pandemic, which is just behind us. And there's been lots of information in the news around vaccine development, which is good, I think, and much needed. The topic for today is very specific and on how you can bring your vaccine to the clinic, into clinical research. In other words, moving it from the preclinical stage to the clinical stage. And we're very happy to have a really very expert and very knowledgeable trainer and speaker on the topic, Joris van der Putte, who is who's had many years of experience in this field, precisely in the preclinical and what is needed to bring vaccine to the market. For instance, he has been like foundational for the IABS, the International Alliance for Biological Standardization, and he has been working across industries, all in the vaccines area. And with that, I will not take any longer. There is opportunity for you to ask questions. If you have ongoing questions, you can put them in the chat. We will address them at the end. If you want to ask your question live at the end of the session, you can also do that. So and with that, I'd like to hand it over to Joris. So if you can take control, Joris, the stage is yours.
Thank you very much, Benedict. It's a pleasure for me to give this introductory session about preparing a vaccine candidate for the clinic. And by clinic, most of you know where the clinic starts. It starts at phase 1 in human health. So I will focalize on human vaccines. That being said, I have been active on both sides of the One Health, in the human and in the animal health side. So when we start to develop a vaccine from start, it's extremely important to have an overview about the whole value chain. It starts, of course, like many of you might know, with the isolation of an agent that provokes disease, that provokes trouble. And then you go to what we call scaling up, trying to produce, to reproduce, then to preclinical in animals. And then if you have a complete file, if you are satisfied, then the whole clinical part, phase 1, phase 2, phase 3, and hopefully sufficient data to write a complete market authorization file and hopefully market authorization and launch. Why do I say that? Because it's very important to be aware, at least for those who are in clinical, what will be required during the clinical phases in order to anticipate and to integrate it as early as possible, as we will see. So I click here now. Yeah, okay. So the preclinical, it can take more or less 3 to 5 years in the clinical vaccine and in the classical vaccine development. And the correct anticipation, as I said, will allow you to save time and money. So overall, for those who are not familiar with the vaccine world, it takes between 5, 6 to 10 years from start to bringing a vaccine to the market. So let's start with the bright idea. You are extremely happy, you isolated a new virus, and of course, you want to develop and bring a vaccine to the market, let's say not next week, but next year. As I said, until now, it was more or less a decade's time work. So when we have our antigen or our pathogen, and if we want to develop a vaccine, it's extremely important to pose some basic questions. Those of you who are in the field of vaccines, they know all that. I excuse myself to be a little bit simple, but I think it's important to come back to certain basic points. Is there a need for a vaccine? This is not an easy one. Need for a vaccine depends on, does the agent provoke disease? This can evolve in time. Is it a vaccine that touches humans, animals, or both? This will already impact the way how you will start the development of your vaccine. In order to develop a vaccine, you all know, or most of you are aware of the huge money investment in addition to personal investment it needs. So therefore, it's important to have an idea about the economic impact of the disease in order to justify the need of a vaccine and, of course, also the market value, and to find investors that will be willing to invest. Having a bright idea can make people extremely, how should I say, ambitious and extremely excited. So I wrote in red no communication because today patent protection is extremely important in order to protect you and in order to create value. A small example I lived personally some decades ago. It was about a very innovative way of administration of a vaccine. And some people were so motivated about that in a small circle, in a region of Europe, I will not tell where, they talked about it. And by accident, 2 journalists of a local newspaper were there, and they wrote about it in a very regional newspaper. When we then tried to patent that procedure, then this small article, only 10 lines came up. So if you want to protect your invention, or when you think it's an invention, silence is gold until you have the official registration that your patent must not be allowed, but that it at least has been registered. The benefit risk, what do we mean by that? So benefit risk, those who are in the field know that the regulators look at benefit risk, and especially governments look at benefit risk in order to know will we be able to fund or pay for that vaccine or not. For your investors, this is extremely important. And it is important that given the complexity of vaccine development, you need the right people, the brains, in order to do it, and already foresee with whom outside your organization you could have to work. Not to forget, it's never too early to take contact with the regulators in order to pose some basic questions, as we will see. So the next slide. So then I think, yeah. Then we come to some very simple questions, but which unfortunately are only posed too late, as my experience shows. Is it a vaccine for all ages? And if yes, what about children? Or is it only a pediatric vaccine? Is it seasonal or not? Is it a One Health? Is it, for example, a mosquito-borne or an arbovirus-borne disease? Will it be a vaccine for travelers, global or regional? And will the market be public, private, or both? And last but not least, are there already people who have launched such a vaccine? What can we learn from those who have launched the vaccine, or will I be the first to be present on the market? Those different, the answer to those different questions will influence what we call your target product profile. I will recommend everybody to start writing it very early, because this is an evolving process. You can decide, for example, that your immunity will take, I just invent something, 1 year, but it can come out that in preclinical and then in first phase that it may be 5 years or only 2 months. So this is something you will have to take into account. It's very important to remain scientifically extremely severe, stick to the data, but sufficiently flexible to adapt according to scientific data. We advance now, and now we have our virus for, let's say, disease X. Importantly for all of you is to know the pathogenesis as much as possible of this disease. When it's an old disease where there is no vaccine still, or there are already vaccines, normally the pathogenesis will learn you, is it a parenteral vaccination or an oral vaccination or a topical vaccination? Will it be live or not? If it's a new disease, and let's use COVID as an example, well, it's not that easy for example, in February 2020, when 3 types of vaccines were developed already, to know will this remain a respiratory disease and will it have an impact? So these are all things that are extremely important and which will determine the profile of the vaccine that you will develop. Let's take, for example, as an example to COVID, but there are hundreds of other examples. You have the virus, and then in order to proceed, you must produce or reproduce your virus if we stick to the classical methods of cell culture. I will not talk about production of eggs, comparing eggs with cell culture. This needs another explanation of about 1 hour, but let's keep to cells. And I don't go into the details of cell lines, etc., and primary cells. That's also another discussion. So we know that in order to find the right cell line, it can take a lot of time, or you can be lucky and during a week you see, well, it replicates, it multiplies. But then you must know, and this will take at least a couple of months, does it produce sufficient quantity of antigen? And does the antigen of the virus that it produces, is it corresponding and still the same as the origin of the disease? The choice of the cell will be extremely important, not only for production and quality purposes, but also, and those who are in the field of vaccines know, if you use an approved cell line where already other vaccines have been produced on, for the regulators it's much more easy, they feel comfortable. If you start with a new in-house developed cell line, then you will have to prove the same quality. And this is also a complete information and discussion of a couple of hours as well. Do I go for a live or an inactivated vaccine? not always easy with a new virus or a new disease, antigen. So I will a little bit come back to the live versus inactivated. This is for the production of vaccines on cell lines, which are still very valuable. Also in the field of COVID, there have been some successful developments on cell lines. And while have they been successful and in a relatively short time, but longer than what I will present next, the platform technologies, because there is decades experience using these cell lines for other viruses or viruses which were or are cousins of COVID, for example. The newest technologies, these are platform technologies, which allow much more an approach vaccine development by design and also quality control by design. Let's look at the practical production system here, which is represented. You have your virus, you start with producing your virus, hopefully in a cell line. You test in general different cell lines, you find one in flasks, and then you have to increase the volume. We call it the scaling up. You do it first in 10 liters, 50 liters, and this can go to 10,000 liters fermentations. However, this can take time if you are not lucky. On average, 1 year is already very good, but this can take a couple of years. When we go to the newest technologies, by the newest technologies, I mean DNA platforms, RNA platforms, and antigen production. I do not say virus production. I talk about antigen production in baculoviruses and insect cells, for example, or in bacteria or in fungi. So this is totally different. You determine the genome of the virus, you cut that piece that you will use because you believe this piece of genome codes for the right antigen, and you insert it in your DNA platform or your RNA platform or your baculo, for example, or your fungi or bacteria in order to produce the antigen in one way or another that you want to be produced. So I will not go into the details of glycolization, etc., having the right antigen. This is part of additional discussions and presentations, of course. Today we can, as COVID has shown, determine the genomes by in silico technologies. Those familiar know this is determining the genome with a lot of mathematical models, very precise, and have the genome in a couple of days. I say 24 hours because in a previous project where I worked on a European level, we have done that. So it's very efficient to very quickly know the correct antigen that is needed for protection, immune reaction, etc. And in COVID again, we were very happy that the Chinese colleagues produced the complete genome. I think it was on January 10 or 11, 2020, and already at the end of February, we were informed about the front-running projects that would make it, and they made it. So they made it thanks to platform technology. For example, you have your target pathogen. The genome sequencing can be done automatically with huge performant models. For messenger RNA, for example, then you translate your RNA piece into a plasmid in order to be able then to in vitro reproduce huge quantities of the messenger RNA, which then you will couple to the lipo nanoparticles in order to be able to vehicle your messenger RNA into the cells and through the lymph node, then go to the lymph nodes and generate the production of antigens and put into place the whole immune system to generate immune reaction and protection. This can be done in a relatively short period. So some of you might know the ambitious CEPI project, the 100 Days project, because this is based on those new technologies, whether they are messenger RNA or another one I will show. I think this is really feasible. And what we have seen in the COVID field, whether it is the recombinant DNA and adeno or the messenger RNA, we have some excellent examples on that. That being said, these vaccines have not fallen from heaven in 11 months. It's extremely important that all of us, we explain and are aware of this is the result of 3 decades work. In the messenger RNA field, for example, there were already 8 million people that had received on experimental basis for cancer vaccines, for example, and others, had received somewhere in the world a shot of messenger RNA, experimental RNA vaccine, which allowed to know a lot. The same is true for the adenoviruses. The adenovirus vectors had already been used in a lot of experimental designs, and not least the experience with Ebola in Africa had helped a lot in order to establish the one of the adenovectors, and the ChAdOx, it was the same. It has been used in a large number already. So we had a lot of experience. But again, this quick development is the result of 30 years very excellent research worldwide. We should not forget that. An alternative in platform technology is the production of particles. The example I give here is the particles based on scaffolding. These are relatively small molecules where you fix a glue and which will be used in order to carry the antigen. So you can produce your scaffold, your multimeric scaffold particle. You can produce it separately, for example, in coli, let's say in the north of Europe, and then you will produce your antigen somewhere in the south of Latin America. Again, this can be done in baculo, for example, or coli, or can be done now. There is a lot of progress in fungi, the C1 fungi, for example, and you can also glue that and bring the two then together in Africa and have your local production there. So this has been done in a European project and shows that it can be quite efficient. So just as an example, like the messenger RNA or the DNA, those new platform technologies are very versatile, which is extremely important to adapt, to be able to adapt your vaccine to new strains and to use the same platform technology also for new diseases. So the decision you will have to make is very important. Which kind of platform? Either the classical cell line, or do I go for new platform technologies? And it will depend a lot on the history of the institute you work in. If you have a long history with cell lines, and if you have a long history of quick reaction with cell lines, I can imagine that the switch to, for example, particles or messenger RNA can be rather difficult because you are comfortable and you know exactly what has to be done by the classical way of cell cultures. There is nothing wrong with them, and we see it now every day. On the other hand, we have seen the advantages of the platform technologies like messenger RNA, like recombinant DNA. A little bit comparison about the development in recombinant vectors. Recombinant vectors are, of course, live vaccines, and you have to pose the question whether you will be confronted or not, which can be unfortunately the case, to the GMO regulation. But here again, that's another discussion which would lead us too far this afternoon. Then once we are here with our bright idea, we must think about already here, anticipate the chemistry, manufacturing, and control requirements. The beauty of this platform technologies, at least in my opinion, is that you can not only create a vaccine by design, but also, since everything is quite well defined, you can also progress quite quickly with your in vitro control, which is the case for many COVID vaccines, for example, because everything is well defined. So today, I would say, till a couple of years ago, your quality control for batch release and in-process control very often had to use quite a lot of laboratory animals. Today, we can get rid of them, at least for the quality control and consistency part, which is a huge advantage and cost saving. So the quality attributes will play a very important role in the setting up of your different control methods. I recommend very strongly to include the GMP and GLP, good manufacturing, production, and good laboratory process principles as early as possible, in order to avoid that you unjustifiably have to redo things you have done before. So I give now a little overview before we go to the scaling up and the preclinical part on animals. So let's use the COVID example. We know it's coronavirus. There are about 50 coronaviruses we know in both human and animal health. And we know from all of them that the spike, you see the spike, is the part which is extremely important to attach to organs, to cells, and to generate the pathogenesis. That's why the publication of the genome was so important, because based on the knowledge we had on coronaviruses, whether it was SARS-CoV-1 or MERS or other coronaviruses, it was found immediately that the genome that was produced, we had to cut where the spike was had its information. Let's now look at the platforms themselves. I make a differentiation between the classical platform approach, that I mean a viral vector like adeno, like pox, and like many others, like also measles. I consider them as classical because you have to go to cell culture in order to produce them, scale up, etc., etc., etc. The advanced technologies, these are the DNA technologies, the particle technologies. The particle technology, we just express the antigen that we need, or the RNA technology in general, but that's not a general rule. They can be quicker. The beauty of those platform technologies is now that there is a huge evolution, not a revolution, a huge evolution from the regulatory part. This has already been done in the new regulation for veterinary vaccines, where once you have established your platform, you do not have to redo all the work you did already on safety, on production, etc., of your platform, when you want to use that platform for a variant or for another disease. You just have to complete your work. I simplify a little bit, oversimplify for the sake of time, but that's the principle. You do not have to redo everything. You can focalize yourself on the specific work you have to do for the new virus or the variant or whatsoever. Let's come now to the stage where we will try to produce, and not only produce, also to fine-tune the control methodologies that we will develop. So again, I cannot insist enough about the importance during the previous stage, the importance of all the decisions you make. Those decisions will make you gain years or you lose years, because once you start to upscale, it needs upscaling most probably in a GMP. You will invest. You must assure that your quality attributes start to become in process. It goes all in parallel. And for the live attenuated vaccines, whether it's adeno or whatsoever, it's highly recommended to assure that your live attenuated vaccine is stable during 10 passages at least. For those who are not familiar, remember I showed in one of the first slides decision live attenuated or inactivated. Live attenuated, it needs in general 50 to 100 passages in order to sufficiently attenuate a virus. There can be exceptions in positive or negatives, but each passage you must consider 1 week. If you go for a live attenuated vaccine and you will need between 50 to 100 passages, that means you have already started for 1 to 2 years before knowing what you will have as kind of vaccine. And then you have to prove that your strain remains stable. That means it has not become virulent when you passage it, and it keeps its immune potency. For an inactivated vaccine, the challenges are different. In general, the immune genesis generated by an inactivated vaccine is different. And there are advantages to take an inactivated in some cases and alive in other cases. I cannot foresee that here. It depends on the disease, the virus, etc., etc. So, but it's all very important to think about that quite early. Importantly, not to forget, and for scientists, this is from time to time quite difficult, write down everything in laboratory books. I cannot repeat that enough, because once you will have to make a market authorization file, the regulators will very much like to go back to trace back in order to be assured when they give the approval that everything is based on science. So the robustness and consistency of the production will intervene here at that stage when you go from small runs to higher runs. And again, the quality by design and the new technologies allow us a very much precise quality attributes control than previously, when we had, for example, to do almost everything on laboratory animals. Another point that can be of importance is where will I develop and where will I produce and where will I go for market authorization, because there be some specificities. So the decision on technology again is extremely important, and again here I would highly recommend to dialogue with the regulators. The regulators are interested not only to know what happens, but to give their input. And for you, it's important to know where the boundaries are, which is not also self-evident when you read the texts. Consistency of the methodologies, toxicology will have to be done also in this phase, linked with the preclinical on animals. And for toxicology, it's extremely important that when you use the toxicology results in your files, these have been done by certified laboratories. Now let's come to the last stage of my presentation before we go to the clinical stage, phase 1. So on laboratory animals, you will have to finalize, of course, the toxicology studies. Toxicology studies must be done at the maximal dose, but when will you decide about the maximal dose? Well, the maximum dose you will be able to decide when you will have done quite a lot of, in parallel, animal models and animal models in order to know from which stage is it maximum and where is then ultimately, in order to generate an immune reaction, the minimum. Determining the right species in order to generate sufficient immune data and efficiency and efficacy data is not always easy. And in addition to that, even with the best model, it's not always easy what will be the relation or correlation, what you see in an animal model. Let's take COVID now, but there are more difficult examples than COVID. when we look at hamsters and non-human primates, of course, when this was done initially, it was not self-evident that the relation with what could happen in humans would be extrapolable. However, when we look at what has been done in COVID, when we look at the results in non-human primates and hamsters of different vaccine candidates and publishable results that were available early 2021, here we can see, for the COVID example, we can see a very good confirmation of what had been seen in the field about the efficiency of the vaccines. So those escalations are extremely important in order to determine your immune profiling of the vaccine, and also important when you do vaccination challenge studies, which are almost always done in the preclinical part. They can learn a lot and give already good indication whether or not it will be possible to work with correlates of protection. Of course, again, this has to be confirmed in humans if the target animal, sorry for that, is the human. So of course, it's not new. You will have to not only look at seroneutralizing or other antibodies, the cellular component of the immunity is also extremely important, like we see now with the COVID, where the CD8 seems to play a very important role in clinical or severe clinical protection. Again, I insist here that the production system must be closer and closer and closer of really GMP. Because when, for example, you will start quite extensive animal models, and if you want then to use these results, when these experiments are done with a vaccine that's only more or less defined, and if on top of that you do not have the history of quite a lot of components of the vaccine, that means in order to write a valid authorization file, you will have to start again. And unfortunately, this still happens too much today. So it's, as said, very important and essential to establish a very good safety profile in animals. In the preclinical trials in animals just before clinical phase 1 allows you to fine-tune still, because there you will see whether your apprehensions on doses, high, middle, low, etc., will be confirmed or not. And this gives you the time in order to validate everything. Then the in vivo immunogenicity and efficacy in a correct vaccination challenge model, I repeat, is extremely valuable for the immune parameters and to see whether protection can be linked to correlates of protection or not. This is the case for some vaccines. This is not the case for other vaccines. But how more we dig into the results and we dig into the different components of the immune system, we see that for a lot of viral diseases, at least, the correlates of protection can be a very important point for the development and the quicker development and the quicker availability of new vaccines. So the quality control in process to assure the consistency, to assure that the 50th batch corresponds with the original batch where you did all the safety, efficacy, or the original batches that you used in phase 1, phase 2, and phase 3, that they all are of the same quality is extremely important. And therefore, in vitro testing really allows you to go quite far in order to determine the quality of your vaccine, whether it's in process, and I repeat, whether it's on batch release. When this is all very well done, I would say you are ready for the clinical trial application. So the first in human clinical trial or the phase 1. And not to forget, you are here at the confirmation of your proof of concept. Investors will carefully look at all these results in order to decide whether they go for the huge investment. As you know, the clinical trials, you are quickly at 100, 200, even 500 million euros or dollars. Very expensive, but these decisions will depend on the quality of your preclinical file and also the assurance that there is really a market need. So I will stop here to give, and I hope that I gave you a little flavor of the interesting and passionate world of preclinical development of vaccines. Of course, each of the 3 steps needs further digging into more information and more discussion in order to have a complete and comprehensive overview. I thank you very much. Benedict, we don't hear you. Benedict, you are on mute.
Sorry, it happened to me again. So thank you. I was thanking you, Joris, for your valuable insight here. And as indeed, as you say, all these topics, they deserve more lengthy discussion. They need more lengthy discussion, I would say. But at least it gives an overview of what it takes to bring the vaccine to the clinic with the quality desired and with the targeted product profile in mind all the time, as you say. We have some time for question and answers. So I don't see any question in the chat, but I'd like, well, to ask the audience who is online. If there's any question, feel free to unmute yourself and raise it here. I don't know, is there any question from the audience? I know that Joris is very well done, has done a great job in explaining, but I'm sure that you must have questions somewhere and somehow.
I have a question regarding in silico studies and the pharmacometrics, so population PK and PBPK studies. Is there any chance that these kind of studies could play an important role in the preclinical development of a vaccine?
That's an excellent question. Indeed, this in silico studies. So to go a little bit in to work with an example. So you isolate a virus, and one of the classical models to go for in silico is, of course, screen the whole genome of the virus, but also to go into to inject the virus in experimental animals. For example, dromedaries, but not dromedaries, alpacas, and so on, where you have these special antibodies that are generated, thousands of copies of different antibodies specifically destined to the different epitopes, and then reversing the information of the antibodies into genomes give you very quickly the result of the not only the genome, but the confirmation where the specific antigens and the important antigens can lay. This has been done, for example, for MERS, and this has generated very valuable information also for the further corona work. So the result of this, if there is no question in the meantime, is that these new methodologies are extremely helpful to determine the right epitopes that must be produced. Previously, this was really much more trial and error. So where you hoped after a certain passages and a certain trials in laboratory or target animals in animal science and animal health, that you hope, you crossed fingers, this will work this time. Now today we can, using the generated antigens that can be produced also on a small scale in other laboratory animals, to confirm that there is a real match between the antigens that is for the antigen that is foreseen and the epitopes, and with the antibodies that are generated, that there is really a neutralizing capacity, for example, and even further down the line, cellular immunity, etc., etc. This is a component which allows us for new viruses, and also in the One Health system, when we want to strengthen the early warning systems in order to test a variety, hundreds or thousands of viruses much more quickly than we could do till very recently.
Thank you.
I hope this answers your question, Leonor. There is, in the meantime, another question that came up in the chat, and the question is, where can we find more information on which animal model can we use for which platform?
Okay, that's also a very valid question. So I would say the animal model is not linked to the platform as such. So first of all, the question we always have using an animal model, and SARS is a very good example again. So initially everybody went to non-human primates, and suddenly it was discovered that hamsters had a pathogenesis which is quite close to what was seen with the Wuhan and the early strains. So the hamster became a favorite model. Whether you produce your vaccine in cell culture or with DNA or with messenger RNA or viral particle, the hamster has been shown to be an extremely interesting model. And it's also used now, I have seen studies also done with antibodies, monoclonal antibodies against SARS. And hamsters for certain strains of SARS, they are extremely ill, and for certain strains at least, they represent very well or predict more or less very well what happens in the severe cases in humans. So that's a quite difficult one, and I should not link that to a certain platform. The objective of finding a good animal model is to find the right immune genesis or immune pathogenesis, which corresponds with what happens in humans. To come back to animal health, it's much more simple, because there you go more or less directly in the target animal, whether it is a chicken, whether it is a dog, whether it is swine, whether it is bovine, etc., etc. There you test really the virus on what it does, and you can go much quicker. In humans, what it's not that self-evident. That's why probably you know that in humans there is a lot of effort done on CHIM, the controlled human infection models, which have been done for RSV, which have been done already for norovirus. The cholera virus is a typical example that of a vaccine that has been produced by just by human challenge models for flu, etc., etc. And now our British colleagues had a very interesting study published in Nature not so long ago about the CHIM about the SARS-CoV-2 CHIM with only 10 viral particles, which is very, very low for the virologists among us. They know with only 10 viral particles, they could really well reproduce the excretion profile, which is of course very important in the epidemiology in human challenge models. So I would say it's not linked to the platform, it's linked to what is doing a vaccine.
Right. Mm-hmm. All right. So there's one, it seems that we answered the question very well, Joris, thank you. There is another question. What are the preclinical or early phase clinical concerns when a company decides to mix the vaccines during dosing or administration phases?
What do you mean by mixing vaccines? It's a homologous and heterologous vaccine, or live followed by an inactivated, or an inactivated followed by a live, or a messenger RNA followed by an adeno, or an adeno followed by messenger RNA, I suppose. Is that correct?
Eva, feel free to interact if you want to more specific, make your question more specific.
No, it's in the corona vaccine phase where a company, where people got, for example, a vaccine from adeno platforms, got a vaccine from mRNA platforms. So those kind of mixing during administration.
Okay, but I think it's not mixing. So you do not administer the two vaccines at the same time.
Yes, it's during the administration in the market for the people. So well, first dose, in the dosing regime, they can mix two different vaccines, right? The first dose could be from one company, and the second dose could be from another company and another platform.
Yeah, so the mixing would be most probably you first vaccinate with, as has been my case, with the adeno, and then you have a repeat vaccination with the messenger RNA. Well, this will not be necessarily part of the preclinical. This will already be part of phase 1, phase 2, even phase 2, phase 3, where and also phase 4. And this has been done in phase 4. We should not forget that these vaccines after 11 months were already on the market, so not everything could be done. The most sensible and most needed things have been done in an extremely professional way, and I want to stress that here, thanks to the collaboration of everybody, the industry colleagues, the regulatory colleagues, who set up this rolling review, who accompanied the development, the science, who accompanied the development, allowed us as a global community to have this initially 3 and then 4, 5 vaccines very quickly, which saved, by the way, I want to insist to that. Some people do not like to hear that, but that's not a problem, that we saved millions and millions of lives. So that being said, it has been shown, particularly a lot of work has been done by our UK colleagues by mixing first adeno, then messenger RNA, first messenger RNA, then adeno. So this has been done during clinical phases, and the result was in fact very good. So that it seems you widen up a little bit your immune spectrum in this combination. So different to that is, and this is an important question, heterologous. Heterologous, and this is not linked to COVID. This is linked to viruses which vary a lot. So I will use strain A and then strain B, and this can become the case also for COVID. Heterologous vaccines, there are examples, particularly in animal health, where the use of heterologous vaccines really increases your efficiency of the vaccine program. But here again, we have to look at disease per disease, virus per virus class.
Mm-hmm. Yeah, absolutely. Well, thank you for that, Joris. I think we've covered all the questions, and I see that it's also close to 5, and I don't want to keep the people longer than needed. Well, let me thank you again for this very interesting and insightful session, Joris. It doesn't need any words anymore that you are really the expert in the field here. I wanted to thank you for that. The reason why we do this session also is to find out if they would be interested in a more in-depth course on the matter, on the topic. So actually, and my colleague Andre will launch a poll right now because we would like to find out from you if you think it would make sense to organize a more detailed session, of course, where more in-depth discussions could take place, even with exercises and things like that, because yeah, we all know that the learning really starts to happen when you really apply things and you have to do things yourself. So feel free to answer this question. We're only having one question here, but we may follow up then with you afterwards, so that we can also make sure that, yeah, if we develop such a course, it is in line with what you would need, basically. So thank you again for taking the time to respond to that question. And well, last but not least, maybe if for any reason you would have any question around the topic or in general of around what ECCRT is doing, feel free to reach out to us anytime. You see our details on the screen right now. You can also leave us a comment at Google. So if you have any questions, feel free to reach out. And we would like to end this session here for this afternoon by again thanking Joris once more. And last but not least, also thanking all of you that have attended the session for being here and like for the questions raised. I think it clearly shows that it did cover a need here. So thanks again. Have a good afternoon, and I hope to see you soon in a next session.
Thank you. Bye-bye.
Bye.